And our first presentation is from Dr. Joshua Gordon, who's the director of the National Institute of Mental Health. He is also the NIMH representative on the AMP Schizophrenia Steering Committee. And we'd like to share his pre-recorded presentation with you.
Hello, and thank you for having me. I'm sorry that I cannot be there in person, contemporaneous with you, but it's my pleasure to introduce this first AMP Schizophrenia Investigators meeting. I am the director of the National Institute of Mental Health, Joshua Gordon, and I wanted to give you a bit of my take on the importance of AMP schizophrenia, and it really starts for me anyway, back in 2013, while on the faculty at Columbia. I remember seeing this in the New York Times, an editorial by Richard Friedman titled "A Dry Pipeline for Psychiatric Drugs," in which he highlights a series of, quote, failed clinical trials in which novel antidepressants and antipsychotics did little or no better than placebos. The company seemed to have concluded that developing new psychiatric drugs are too risky and too expensive. That was the story in 2013, and it continues to be a bit of part of the story today. A lot of for-profit entities have left the CNS or decreased their investments in understanding and treating psychiatric illness. And this was a disturbing trend for those of us who believed that there were better treatments available for individuals with schizophrenia and other serious mental illnesses. About that same time, in 2013, my predecessor, Tom Insel at NIMH, was working with an early brand new program that the Foundation for the National Institutes of Health came up with. It grew to be called the Accelerating Medicines Partnership. At that time, it was called a Target Validation Consortium, but the idea was to put together what you all are now participating in, public-private partnerships that would work in the pre-competitive space to identify novel targets for treatment and novel approaches to testing those treatments. One of the original work groups in that planning process focused on schizophrenia, and it included Dr. Linda Brady, the co-chair of the AMP steering committee, as well as members of academia and the private sector. They came up with a project not that different from the current project, and proposed it for funding on June 28, 2013 to the group that was forming the AMP process. And unfortunately, at that time, it was decided not to move forward with this project for a number of different reasons, including the fact that it was challenging, or thought to be challenging, to bring the private sector partners on board. After that disappointment, we put a little bit on the shelf at NIMH, the idea of forming a public-private partnership in psychiatry, and moved on to other endeavors to try to enhance our research portfolio and development of novel therapies. Now, one of the reasons why we saw this exodus of drug companies from the psychiatric space is this idea that the neuroscience that we had been doing and the clinical science that we've been doing had not resulted in effective new therapies, and that therapy development was really a game of chance. And I just want to point out that since 2013, there have been a few developments. I will highlight one that suggest we are in the infancy of a true revival, or not revival, a true origin, a new way of developing psychiatric drugs that does build off of science, and in particular builds off of basic science. And that era was heralded a couple of years back by the FDA approval of the first treatment for postpartum depression, brexanolone. Brexanolone is a drug, many of you may know, that is a positive allosteric modulator of GABA receptors. And it actually is a synthetic formulation of a natural compound that we have in our brains, allopregnanolone. And this drug has been shown to rapidly reduce postpartum depression symptoms in seriously ill postpartum in seriously ill women suffering from significant postpartum depression, and it can help women within a matter of hours and last for a number of weeks. But what many people don't appreciate is the role that basic neuroscience played in the development of this drug, and in particular, basic neuroscience either carried out at NIMH or with NIMH funds. So I want to highlight that for you today as sort of a background for thinking about why we have the ability to do something now in the psychosis space. So as many of you may know, NIMH was established in 1949, and soon thereafter, one of the original recruits to the NIMH intramural program was a brilliant young scientist by the name of Julie Axelrod. Julie, over the subsequent decades, really delved deeply into the neurobiology of neurotransmission and developed a sophisticated understanding of many of the neurotransmitters that we know today, how they work, what receptors they bind, etc. And for that, he was awarded the Nobel Prize in 1970. Why do I bring Julie up? Well, really, his work actually forms the foundation for much of what we understand about many of the drugs that work in psychiatry, but also he had a number of acolytes, a number of mentees, one of them by the name of Steve Paul in 1986, along with several others at NIH and NIMH, went on to study a particular compound noticed in the brain called a neurosteroid or actually a series of compounds, neurosteroids, and one of them allopregnanolone, to show that these neurosteroids, sorry, I'm going too fast, these neurosteroids modulate the GABA receptor, which of course Axelrod and many others had shown was the predominant inhibitory neurotransmitter of the brain. This is a work by Steve and colleagues published in Science in 1986, showing responses to GABA over time, to spritzing on GABA onto neurons over time. You can see the inhibitory currents invoked, and then you punch on a little bit of the agonist, and it's an increase in those currents. You can see that even better in the blowups down below. So this steroid hormone modulated the GABA receptor, enhanced GABA currents in neurons. The other thing that investigators then did a little bit later in 1991 was show that this compound, allopregnanolone, was synthesized or synthesis of an allopregnanolone was increased in response to stress. And that's shown here in a set of curves of allopregnanolone levels after a stress induction in an animal. I believe it was a rat, showing increases in cortex, hypothalamus, and even in plasma that one could detect. This led to the theory that allopregnanolone and its modulation of GABA receptors might play a role in the stress response, and indeed might play a role in stress-related illnesses such as depression. That theory underwent a lot of different confirmation in various preclinical studies. But nonetheless, it was still open to debate as to whether it was causally involved, for example, in adaptive or maladaptive responses to stress. But one other theory arose a little bit later in the early 2000s, and it was predicated on the notion that allopregnanolone was actually synthesized out of progesterone. And recognizing that progesterone, of course, crashed and decreased dramatically in concentration with delivery, the theory was made that in the postpartum period, with the dramatic decreases in the levels of progesterone, there would be similar decreases in the amount of allopregnanolone, and that could in susceptible women predisposed to postpartum depression. A number of preclinical studies, as well as a few clinical studies, gathered some data in support of that hypothesis, leading to the notion that perhaps allopregnanolone or analogs thereof might be an effective treatment for postpartum depression. And although that process from theory in the early 2000s to phase 3 trials took a while, in 2019, brexanolone was, as I showed you, approved by the FDA. Now, of course, you know, I've sketched out this theoretical mechanistic pathway. We don't really know all the details, whether all the details of that mechanistic pathway are true, but nonetheless, this basic science process, identifying a compound in the brain, understanding its mode of action, studying its regulation and its synthesis, led to a hypothesis about clinical efficacy that, when tested, actually worked. And we now have a brand new treatment for postpartum depression in brexanolone, as I said, a formulation of allopregnanolone, as well as a number of small molecule modulators of GABA receptors that are currently in clinical trials, as many of you may know, for postpartum depression and for other forms of depression. So that's the bed-to-bedside story that breaks the mold, that paves new ground in terms of the possibility that scientific understanding can actually lead to novel therapeutic approaches for mental illness. And with that process well underway, with the interest in small pharmaceutical companies like Sage Pharmaceuticals, the company that brought brexanolone to market, we wondered, along with many others, whether another chance at a public-private partnership might be possible. Now, it probably wouldn't have gone any further than wondering if it wasn't for, as many of you know, the National Alliance on Mental Illness, working together with the Stanley Center at the Broad Institute, who in a meeting of like minds decided that they wanted to try to bring back some interest on the part of pharmaceutical industry in delving into serious mental illness in general and schizophrenia in particular. They started a series of meetings that occurred in the year before my arrival at NIH in 2016, and then continued after my arrival. And recognizing that we had made a lot of progress in the biology of schizophrenia. We had, as shown in this upper right-hand corner of the slide, shown that actually aspects of the dopamine hypothesis, believe it or not, for schizophrenia are accurate. That is, that there were increases in the binding of D2 receptors in individuals with schizophrenia in the striatum. We by then had 150 or so. Now it's well over 250 loci in the genome that were statistically significantly linked to schizophrenia, providing additional biological clues, several of which here I am showing the tallest peak, the MHC, but several of these led to plausible biological hypotheses of contributing factors leading to schizophrenia. And now that's the biological background, but by then also NIMH had been laying the groundwork in terms of studying the different phases of the illness. And many of you in the room played a role in this process of really trying to understand this early prodromal period of attenuated symptoms that typically, although not always, preceded schizophrenia. And the idea between these biological pathways that we identified, some of which were playing roles at specific neurodevelopmental events, is that maybe one could learn enough about these individuals in this prodromal state to identify potential biological pathways that might be impinged upon by the mechanisms that lead to schizophrenia and test, sorry, I don't know why the slides keep advancing on their own, and test psychopharmacological interventions that would help people in this phase. And from those discussions was born the AMP Schizophrenia Partnership, of which you are a member. So we are charged now, and that partnership involves several pharmaceutical companies, a number of nonprofit organizations, as well as government funding agencies, both here and in Europe, as well as the FDA, which is involved from an expertise perspective, as you all know, in terms of trying to figure out whether we have enough here to work on for either biomarker qualification and or treatment. And you all are members of this truly global network that is being funded by this public-private partnership to move beyond the simple characterization of individuals in this prodromal state, move beyond the hypotheses that you make about the pathophysiological processes that are underlying that prodromal state, move beyond the recognition that individuals in this state have a potential of 3 or more outcomes, that is, return to a healthy state, continuing of some level of psychopathology, and progression to psychosis, to try to identify the map of those pathophysiological hypotheses onto those different states and try to test novel therapies that might impinge upon those pathophysiological processes. That is your charge, and the charge is nothing less than trying to rejuvenate the entire industry's approach to treating schizophrenia. We want to get schizophrenia before it's born. We want to interrupt the process. In order to interrupt the process, we have to understand the process. We made a lot of progress there, but we have more to make, and we need to test methods to try to interrupt that process. Along the way, we also hope to help the approximately 1/3 or maybe more of individuals in that program who will go on to have persistent symptoms of one type or another, but who may also benefit from therapies. So we have a lot of work to do. I really appreciate you all coming together now as the first patients are being enrolled across the globe to look at what you've built so far and to think about how that's going to extend into the future. We pledge at NIMH our support, our best efforts to make this partnership work and to continue it moving forward. And we pledge also to work with you all to make sure that your ideas play the important role that they're going to play in making this future realized. So thank you for coming again, and I'll turn it back over to the chair.
Well, thank you very much for that welcoming from Dr. Gordon.